Hello everyone. In this reel I am going to talk about the free radical, its source and the mechanism of action of free radical in our body. Free radicals are nothing but they are the atoms or molecules that are produced by normal cellular metabolism with one or more than one unpaired electrons in its valency shell or outer orbit. And the example of free radicals are ROS, reactive oxygen species, RNS, reactive nitrogen species, and RSS, reactive sulfur species. This means free radicals are those atoms which have unpaired electrons, and this unpaired electron will always search for one another electron and to become paired and stable. So for the stabilization they will snatch or they will get the electron from the other compounds, and this will cause the oxidation reaction. But free radicals have some beneficial role in our body. It functions in the immune system and also in cell signaling pathway. And in excess, they will harm the DNA and also the cell membranes, and they are carcinogenic in nature. Some examples of ROS and RNS are written. The example of ROS are superoxide, hydrogen peroxide, hydroxyl radical, singlet oxygen, and RNS examples are nitric oxide, nitrogen dioxide, nitroxyl, and peroxynitrite. In this video, I am going to discuss about the mechanism of action of lipid peroxidation. Lipids are mainly present in the cell membrane, and the cell membrane of our cells are mainly composed of by two components, that is a protein and lipid. The major part is lipid, and this lipid contains PUFA, and these polyunsaturated fatty acids are very prone to oxidation. So free radicals can easily oxidize these PUFAs. So here the lipid peroxidation has three steps: initiation, elongation, and termination. So in the initiation step, the RH, that means PUFA of cell membrane, acts with OH, that is hydroxyl radical, and forms one free radical. This is PUFA free radical, okay, and one molecule of water. Then this free radical, carbon free radical or PUFA free radical, will come into the elongation step, and in the presence of oxygen it will form lipid peroxyl. This lipid peroxyl will further act upon a new PUFA of the cell membrane and forms lipid peroxide and one free radical. This lipid peroxide is also known as promoter. It will also initiate the reaction, and this will be continued as a chain reaction. So the termination is very necessary in the case of free radical reactions, and the termination occurs when two lipid peroxyl molecules combines together and one oxygen is released. Free radical reactions need to be stopped, otherwise the cell membrane will be oxidized, and this damage may cause some degenerative diseases. Sources of free radicals are two types: endogenous free radical and exogenous. Endogenous free radicals are those free radicals which is formed internally in our body, and this can be through the electron transport chain leakage, NADPH, oxidase enzyme, and respiratory burst, lipid peroxidation, that is mainly discussed in this video, xanthine oxidase enzyme. And exogenous free radicals mainly contains alcohol, drugs, smoking, and radiation. Those are mainly external factors that is introduced with the body. So this is for today. I hope this video will help you. Thanks for watching.